about how many of you guys are freshmen, sophomores? Freshmen or sophomore equivalents? Okay. All right. And I guess the rest of you are junior, seniors, is that right? Who are the junior, senior? Okay, great. And then among you guys, I know you guys are here because you're interested in STEM. And so I'm also kind of curious to see how many of you are intended or currently in a science major. Okay, a life sciences major, okay? Good number of you. And how many are you in an engineering major? Okay, a few of you guys. And how many of you are in non-engineering, non-science STEM majors? Okay. Okay, great. A number of you guys there as well. Okay, well, I know that in this talk, what I'll basically do is give you a little introduction of how I got interested in the field of STEM. Then I'll tell you a little bit about the research that our lab does at Stanford. And then finally, I'll end up with a few, I think, advice or nuggets of truth that I hope will be useful for you guys as you guys are navigating your career paths. And of course, this is an informal discussion, so feel free to stop me at any time if you have any specific questions. So before I get to this picture, which is maybe dating me a little bit, I want to tell you a bit of how I first, where I'm from, and how life was like growing up for me. Well, my parents are from China. We are first generation. I'm the fourth out of 5 kids. And in fact, I was born about 6 months after my family moved here from China. And in my family, my mother took care of us because there were 4 of us little kids. And my dad basically worked 2 jobs at a time. And growing up was rough. My parents did not speak a word of English. And sometimes they would tell me like, oh, for the longest time, we thought this bottle of floor detergent was the dish detergent. So we used floor detergent to wash our dishes for like a number of years until somebody figured out that we were not reading the labels because we're all my kids were not reading the labels, right? And sometimes I hear stories about how my family, where we grew up in New York, in Brooklyn, and how my dad would basically walk from Manhattan to Brooklyn over the Brooklyn Bridge just to save a train ticket that costed, at that time in the 80s, 25 cents. So to save 25 cents, we would walk across the Brooklyn Bridge with groceries and with everything to and from work to save 25 cents. I would also hear stories about how growing up in our family at that time, at that time, there was 5 of us, growing up there and thinking that my mom had to deal with a $10 was our budget for food for the entire week. And when my parents tell me these stories, I think it really helps me to wake up like, wow, these are some of the sacrifices, these are the struggles that they went through to come to the U.S. here so that their children, us, can have a better future. And I think those are some of the reasons that really fueled me to become, wanting to make sure that I do take advantage of the opportunities that my parents did not have. My parents did not finish elementary school. My mom finished up to 5th grade. My dad went to I think 6th or 7th grade maybe. And knowing that, I think that it really inspired me to make sure that as I grew up, that I would make my parents proud by taking advantage of some of the opportunities that I would have that my parents did not. So that's a little bit about how I was raised in my family situation growing up in Brooklyn, New York. And so one of the opportunities that I took advantage of was back when I was in high school. In fact, I was a sophomore in high school. And that spring, preceding the summer, I was okay, what am I going to do this summer, right? And so what do a lot of people do when they're decided? Well, just apply to a lot of different things. I'm like, okay, whatever I get accepted into, I guess that's what I'll do. Well, that spring, I applied to a number of different summer internship opportunities across the spectrum. I got some art things, some archaeology this, some science this, some business that. And in the end, the one that I got accepted to turned out to be an internship through the New York Junior Academy of Sciences. And it basically matched us, high school students, to labs in the New York area for the summer. And at that time, I was a high school sophomore, and sure, I'd taken some science classes, and science was kind of interesting, but nothing really standing out to me. But I took a chance and did a summer internship and had a very supportive mentor. Really taught me everything. I knew nothing about actually how to do research, how to hold on to a pipette, what do cells look like, how to use a microscope. No idea of any of those kinds of things. And my mentor, his name was Dr. Sunny Luke, and at the time he was a doctor at a local hospital near my house. And I spent that summer with him, along with some other interns. You can see it's the number of us. And this was just a post picture we took to the local newspaper. Certainly would not be this crowded when you're doing research. And basically, through that summer, I got really interested in the area of science. And the work that we were doing at the time was related to cancer, in fact, ovarian cancer, and trying to look at genetic mutations in ovarian cancer. And for someone who was a high school student who knew nothing about cancer, nothing about actually how to do research, that was a really transformational experience. And through the years, that kind of inspirational experience has always stayed within me, and is one of the reasons for why now I'm kind of returning the favor, right? By essentially having my own research lab where then I can mentor the next generation of students. So that's a little bit of how I got interested in science. Was it a little bit of luck? Maybe. Okay, so I mentioned that I was working in a lab that studies cancer and genetic mutations, and those are just images of how chromosomes look like in a cell. And then I went to college, and during that process, I got really interested in something else. It was a field called bioengineering. And at that time, it was a field that was just starting up. And what is bioengineering? Well, an example of bioengineering is this artificial ear that is grafted onto the back of a mouse. Okay, but what really is bioengineering? Now, many of you guys may already know what bioengineering is, but bioengineering essentially is a hodgepodge of lots of different life sciences disciplines as well as engineering disciplines. Bioengineering is really encompassing all of these different life science and engineering disciplines with the goal of integrating principles from each of these different distinctive disciplines so that we can solve complex biomedical questions. And along the way, to solve complex diseases nowadays, we kind of need a multidisciplinary approach. You may need to understand the basics of biology and chemistry, but at the same time, you also may need to know principles of engineering. Maybe you need to know actually some computers. Maybe you need to understand biomaterials. Maybe you need to be good at math and statistics to do some of the work that you do. So bioengineering is a very broad discipline. It basically means incorporating principles from elements of life sciences as well as engineering to solve a complex biomedical problem. So why was I excited about engineering? Well, it turns out that around the late 90s, a specific, back when bioengineering was forming, a specific discipline of bioengineering called tissue engineering was just being born. And I'll talk to you about what tissue engineering is in a second, but before I do that, I just wanted to backtrack, just as I was telling you my sort of my training history, is that I decided to do an engineering discipline as an undergraduate. Initially, I thought cancer was really exciting. I thought I was going to be PNAS. I thought I was going to study cancer and ways to treat cancer. But I got really interested about engineering disciplines because engineering disciplines are really our way of problem solving. It's not so much about memorization. In fact, practically all my engineering classes are open book. You can bring as many notes as you want, open book, no problem. But if you don't know how to solve the question, well, the open book doesn't really help. And so I got really excited about this idea of, wow, I don't have to memorize everything. That's great. And learning how to essentially develop skills for problem solving, that's something that I can take with me the rest of my life. It doesn't have to be related to this major. I can apply engineering disciplines to the way I do routine things, in my personal life, and how I organize my closet, right? I can use engineering discipline to solve problems and apply it to multiple areas in my life. And so that was one of the things that I found really exciting about engineering. But back at that time, bioengineering was, as I mentioned, was just getting started. So most colleges didn't even have a bioengineering major. And so at that time, I chose what was closest to bioengineering, which is a discipline called chemical engineering. So I'm kind of related, but not exactly the same. Chemical engineering means utilizing the principles from both chemistry as well as traditional engineering disciplines like mechanical engineering. And so that's where I was. I did my undergraduate at MIT. And then after that, because I really knew that I wanted to do specifically bioengineering work, because I wanted to really focus on essentially diseases, right, which needs to understand biology. And that's why I did and I knew I really loved research. That's why I did a Ph.D. at UC Berkeley. And then from there, I decided, okay, that's great. I've done a lot of engineering work, but I really want to understand diseases, because I've been really with engineering faculty. I wanted to understand the perspective of clinicians, doctors. And so I did what we call a postdoctoral fellowship at Stanford in Cardiovascular Medicine, where I was able to work with cardiologists to really kind of understand what are the kinds of cardiovascular diseases that they need to treat, what are the problems, what would be potential solutions to some of the limitations that they currently have. And so that's a little bit of my training, and then eventually I stayed on the faculty at Stanford. So that's a little bit of my training history of sort of how I got here. And so as I'm talking about engineering and thinking about academia, I would also inspire you guys to think about academia, meaning becoming a teacher or becoming a professor as a potential career to think about. Now, what would be the reason for why you would want to be a professor? Well, these are just some things that I think highlight what life is like as a professor. Well, we teach courses to undergraduate and graduate students. Besides teaching, we also think about cool research ideas and trying to get those ideas funded by writing grant proposals. We write a lot of those. But we also are treating the next generation of students, graduate students, postdoctoral fellows, and supervising them and teaching them sort of the ways to become an independent researcher. As I mentioned, we write a lot of grants to try to get money to do what we like to do. And then finally, we also give talks and present data meetings so that we can have collaborative exchanges of ideas, as well as to publicize the work that we think are exciting. So this is how life is like as a professor. But what are the benefits of a professor? Why would you want to be a professor? Or what are other kinds of careers? Well, as I mentioned, you can do cool research, right? And specifically, there are cool research that you think will help our world or help humanity. And so I'll tell you one of our craziest things that we've done recently is this. This is actually a write-up of some of the work that we're just starting to do on the International Space Station. So this is something that I just thought of one day to say, hey, wouldn't it be good if we engineered tissues in space? And would someone actually be able to fund that, right? And we were able to get it to work. So actually, in this fall, we're going to be launching engineered tissues on SpaceX to the International Space Station, and the astronauts there are going to help us perform our experiment. And this is something I would have never thought would be possible, right? But, if we can get funded, even things that we think are wacky ideas, we can do. Isn't that exciting? So this is just an idea of something that can be wacky, but to me, I think it's a very cool idea of being able to engineer tissues in space. So anyway, so this is just a little bit of a snapshot of some of the things I think are really neat about being a professor and being able to have the freedom to study whatever you think is interesting, as well as things that we think are impactful. Okay, so I mentioned about doing cool research, right? But importantly, I think that being a professor is not, I don't take it lightly. And the reason is that, one, you're transferring knowledge to the next generation, and you're in a position to influence the students that you mentor, in a good as well as bad way, right? And I remember the times, and I'm sure many of you guys recall some of your favorite teachers, and you probably recall some of like your least favorite teachers, right? So an example for me was in high school, I just had the worst luck of having just really bad teachers in social studies. I had just terrible luck, right? And what did that lead me to the impression of? Well, I'm not interested in social studies, right? And now if I had had a different experience, I had very inspirational, really good social studies teachers, hey, maybe I would have wanted to go more in detail into social studies, but I didn't. And so therefore, I didn't go into that major. And in that same way, I don't take it lightly that being a professor means that you're in a position to be able to make a positive influence on the next generation of students. But if you do it badly, you can also steer them away. And so I think that this is a very important reason for being a professor, to have that kind of positive influence on the next generation. And then I put finally flexible schedule in parentheses. Means that basically that we have the flexibility to control our schedule. Maybe it's not a 9 to 5 kind of day in, day out kind of job. There will be times when we'll be working a lot more than 9 to 5. Other days, maybe less, less hours, right? So in that sense, it's flexible, and that it's not strictly 9 to 5 kind of job. And just as an additional thing that I want to mention is that through some of our interests in being able to promote leadership as well as to promote opportunities for students to learn new sciences and to be able to interact with different scientific professionals. One of the things that I started back in college was a nonprofit organization which is called the International Institute for Scientific and Academic Collaboration. It was really designed to enhance and foster intercollaborative research and learning between various countries. And so I just wanted to mention that as a way to empower you that, just because you guys are students doesn't mean that you guys can, you can do whatever you think is important and makes an impact. If you want to start a nonprofit organization, there's nothing stopping you from starting a nonprofit organization. If you want to start any kind of project, you can make it happen, right? And so there's nothing that says, oh, well, you have to first get a Ph.D. in this, have 10 years of experience in that, and get an MBA in this in order to have an impact. You can have an impact any day that you want, as long as you are determined to do so. Okay, and then finally, I'll just to finish up this, wrap up this part about what is life as a professor. You can tell I'm really trying to sell you on being a professor here, right? Is you have a lot of different hats to wear. One is that you're the CEO, you're not the executive, you're the CTO, you manage all the technology. You run the lab, you determine what's the direction, you call all the shots of the direction, right? You're also the chief financial officer, right? You actually deal with the money, right? And that's why we write a lot of grants to make sure that we can support the students and support the research that we want to do. You're also the head of HR. You're responsible for hiring people, finding the right kinds of students who you think will be successful in their lab. You're also head of PR. You're in charge of advertising, marketing your lab, letting everyone know, hey, we're doing exciting research, collaborate with us. And then finally, you're also the safety officer, right? You make sure that everything is running in a legitimate, safe way. In California, if your trainees or if your students, let's say, inadvertently blow up a lab or something like that, you as the lead of the lab becomes criminally responsible. So that's just another side from what we won't emphasize too much on. And so finally, if you are thinking about pursuing a career in not just in academia, but also specifically in research, right? Because not all professors do research, but if you're interested in being a professor who does research, right? Some of the questions you want to ask yourself are you interested naturally about the world around you and yourself? Are you passionate about searching for knowledge and answers? And finally, are you patient with a capital P, I would say, because research is called research with R-E because you do it over and over and over again. And that's why you really need patience and motivation to be able to go through many iterations of failed experiments in order to optimize the one that actually works for you. So I've talked to you now a lot about what I think is really exciting about academia, about being a professor, about doing research. And so in the next 20 minutes or so, I'm going to tell you a little bit about the research that we do in our lab, and then finally end with hopefully some advice that you guys may find beneficial. So our lab is a cardiovascular lab. So we study cardiovascular tissues. And one of the main tissues that we care about are blood vessels. Everyone's got blood vessels. And the majority of the organs in our body also have blood vessels. And within the blood vessels, one of the cell types that are really important are called vascular endothelial cells. These are cells that line the inner lumen of all of your vessels. And they are essentially responsible to be like Teflon. They're like the Teflon of your blood vessels. They make sure that nothing attaches to your blood vessels. What happens if it does? If junk attaches, what happens? You start to get atherosclerosis, and that's what atherosclerosis is. When different kinds of fats and other lipids get lodged into your blood vessel, and because of that, blood flow cannot go through. And what happens if it happens to the heart? Well, blood flow cannot pass through a specific vessel in your heart, you can get a heart attack, which is fatal. But, and that is fatal, but blood vessel obstructions can happen everywhere else, outside of the heart. And so the endothelial cells are the ones that are most important in making sure that it has a Teflon-like inner surface. Because you don't want it to be like velvet, for example. And when we think about what we call a tissue engineering perspective, we're trying to engineer tissues that look like the organs in our body. We need to be able to engineer artificial blood vessels, which means that we need endothelial cells as one of the principal components of our engineered tissue. Okay, so I mentioned we are a tissue engineering lab specifically. What does tissue engineering mean? Well, tissue engineering over the years has what we call a tissue engineering triad. And the idea is that when we want to engineer tissue, that means we want to create an artificial tissue, engineering, that mimics the structure and or function of its native counterpart. So that's what tissue engineering is. Why do we care about tissue engineering? Well, just to give you an example, think about the number of people who need organ transplants. Let's say the ones that need a heart transplant. Well, it turns out that so far, the best solution is an organ transplant from a donor, like a donor heart transplant. But the fact is that the number of donor hearts for transplantation does not meet the expectation or the needs of those who actually are in need of a replacement heart. And so what happens to those individuals? Well, usually they just won't get one, and in the end, their lives will be truncated because of that. But what if we could engineer these organs from scratch? Maybe we wouldn't have a shortage of organ shortage, basically. And so that's one of the things that we find really exciting about this field of tissue engineering, that we can create an artificial or an alternative way to be able to provide these engineered tissues for patients who need a replacement one. And one of the things that we are really interested in doing is engineering both blood vessels as well as engineering heart tissue. And over the years, it's come to be known that in order to create an engineered tissue, you need at least 3 important ingredients. And the 3 basic ingredients that you need include some kind of cell, because it's biological, right? So it's got to have cells in it, right? And then it needs to have some type of structure. And that's what we call the extracellular matrix. Our body, our cells, are 2-dimensional. The only way that we have any 3-dimensional shape in our cells is because there's a scaffolding structure, which is what we call the extracellular matrix, that gives our organs and our tissues shape. And then there's lots of chemicals that basically tell cells what they need to do. So that's what the soluble factors are. But the idea is that if you can engineer tissues, you can create new heart muscle, you can create new blood vessel, and in fact, tissue engineering can be used to basically engineer any kind of artificial tissue. That's the goal. But now I mentioned the cells part. How do you get the cells then, right? Would you take your own cells and put them in an engineered tissue? Well, in some cases you could, but in other cases you can't just take your own cells and make an engineered tissue from it. One example of it is the heart cells in your body. Did you know that the cells in your heart generally do not proliferate, they don't grow, they don't divide after you're born? And so that's what makes heart attacks so devastating. So when you have a heart attack, part of your heart muscle dies. Those cells die. When those cells die, they should not get replaced because those cells in the heart, which are called cardiac myocytes, the muscle cells in the heart, they do not divide, right? And so one is trying to engineer a biological artificial heart tissue, like, okay, well, adult human heart cells do not divide. How am I going to be able to even grow up enough cells to be able to make an engineered heart to begin with? And luckily, in the past 10, 20 years, pluripotent stem cells have really revolutionized the field of tissue engineering. And pluripotent stem cells are essentially cells that can be derived from the embryo, or more recently, you can derive them artificially using a process called reprogramming. But the idea is that you're able to get these cells that are very primitive, they're very flexible. And these are called pluripotent stem cells because they have the flexibility to turn into almost any kind of cell in the body. So they're not committed yet. And we can essentially coax them to turn into heart cells, blood vessel cells, nerve cells, skin cells, any cells you want. And so over the years, these kinds of pluripotent stem cells have become essentially the source of cells that we use for tissue engineering. So the idea is that we basically commit them into cardiovascular cells using a process called differentiation that says, okay guys, turn into cardiac cells. And then once they do, we can use those cells for tissue engineering applications. And as I mentioned, the endothelial cells, which are really important for our blood vessels, right? So in our lab, we successfully make them. So these are just some images of how endothelial cells look like. So the lighting isn't that great. But endothelial cells are known to have very specific markers that identify them as endothelial cells. And two of those markers, one is called B-cadherin, and another is called C31. But now, in a lot of our studies, we want to be able to track these cells once we implant them into animals. And one of the ways that we can track them is by actually genetically modifying them using firefly genes, in particular, firefly luciferase, right? We know fireflies, they glow, they're bioluminescent. And so we can artificially insert the fluke gene, which is the firefly luciferase gene, into the cell so that they are bioluminescent. And the image up here is just how it looks to show that they can now acquire bioluminescence just like firefly luciferase. And in fact, at the same time, we also genetically modify with what? Green fluorescence protein from jellyfish, right? So they fluoresce in green naturally, and we can extract that gene from the jellyfish and also put them in our cells. And so that's why these cells you can see are naturally fluorescently green, because we've successfully engineered the GFP, the green fluorescence protein from jellyfish, into these cells, as well as the firefly luciferase, so that they're both bioluminescent as well as fluorescent. And so in doing so, we can track these cells in vivo. Okay, so I mentioned before that we want to engineer tissues, right? And one of the things that our lab is most specifically interested in is how do we engineer tissues such that they look like the natural physiology, right? So this is actually a 3D reconstruction of how the vasculature of the heart looks like. And what you see is striking. So what you see in the brown, those are actually the small blood vessels. We call them the microvasculature. So what is striking about these blood vessels? So the big arrow, is showing that they all have some kind of order associated with them. And the reason is that in the heart cells are very much ordered, and these blood vessels also have a defined order to them, right? And so one of the things that we have been really interested in is, well, how do we generate these blood vessels that are composed of these endothelial cells, but then how do we make them have organized structure like this, right? And generally, you can imagine that these basically look like they're parallel line. Now, granted, they're not perfectly parallel line, but generally speaking, you might agree with me that they look parallel aligned. And so, how do we try to make the cell become parallel aligned? And so, what we do is that we utilize scaffolds that give them these kinds of instructive cues that say, hey, organize your bodies along these straight lines that you guys sides look parallel aligned. And so, as I mentioned that the cells in our body all sit on some kind of extracellular matrix, well, the way we artificially do that is by creating scaffolds. And these are scaffolds that are made out of biocompatible polymers. And we can fabricate them in different kinds of shapes and sizes. So you see, they look like little noodles. And in particular, we can fabricate them such that the noodles are all straight, like a bundle of chopsticks, I guess. Compared to what we call randomly oriented scaffolds, where the fibers are kind of what you see, they're kind of randomly distributed. And what's really neat about them is that, and then just to give you an idea, this is how they look grossly. So they're about 7 meter in thickness, right? And we actually, when we make them, we make whole sheets of them. We just punch them out into different sizes. So this just happens to be one particular circular sheet, but we can make them in all sorts of shapes and sizes. But importantly, if we were to see, or basically grow the induced pluripotent stem cells, and just to remind you, the induced pluripotent stem cells are the cells that are over here. So they're pluripotent cells that are derived from a process called reprogramming. So if we were to grow these iPSCs into these 3D scaffolds, the goal is, well, can we coax them with the orientation of those fibers to coax them to say, okay, turn into endothelial cells, and not only that, but align along the direction of those fibers. So basically, those fibers are pretty much, we're trying to use this fiber orientation as a way to say, okay, endothelial cells, organize yourself along that direction. So that is one way, which we call spatial patterning cues, to basically instruct cells and tell them how to organize their cell bodies, right? But the very first thing that we want to do is that when we grow them on these scaffolds, so this is the randomly oriented scaffold, and this is the randomly oriented scaffold, this is the aligned scaffold. And those huge arrows give you a sense of how the individual fibers look. And the cells are just kind of sort of sitting on top of these fibers, right? So the first thing we cared about is, okay, are the cells even alive? And why does that matter? Well, because these scaffolds are very thick. They're about a centimeter thick. And the general rule of thumb is that beyond about 500 microns in thickness, you will reach issues of what we call nutrient limitations. Right? That has to do with the diffusion of nutrients. But these are very porous, and so because of that, we did not really have any problems with cell viability. We use these fluorescent dyes where the live cells turn green and the dead cells turn red. So as you can see, yes, there are going to be some dead cells, but the majority of the cells are alive and green, right? So that was really comforting, right? And we can confirm that the cells are able to penetrate through the entire thickness of the scaffold. So it's as if we cut the scaffold in half like this, and then we looked through the thickness. This is the top face, and this is the bottom face. And we labeled, in this case, with a red dye just to see all the cells. So that we can see that the cells are able to grow and penetrate through the entire thickness of the scaffold. Okay, now getting to the punchline. Can the topography or the spatial patterning of the scaffold do have an impact in terms of how the endothelial cells form their network structure, their vascular structures, right? And so this is an image of what we call confocal microscopy images. But basically, what you're seeing here is a protein that's called CD31 that is a definitive marker of endothelial cells. And so what you're looking at are the endothelial cells. And what I'm showing you here is that on the aligned scaffolds, you can see that the endothelial cells are forming these vessel-like networks that are preferentially in the vertical direction, right? Whereas in the randomly oriented scaffold, you can see that they're pretty much in any given direction. And so we found it to be really important. So I'll just skip over to show you in 3D sort of how these vessel networks look like. So that's on the randomly oriented scaffold, and so the CD31 is in red. So I know it's a little bit confusing to see them moving simultaneously. But the thing I want you to appreciate is that in this sort of 3D rotating view, you can see that those endothelial cells are basically forming vessel-like networks that on the aligned scaffold are pretty much vertical. And that's because those scaffold fibers are also vertical, which means that we were able to successfully give them these kinds of what we call spatial guidance cues that basically tell the endothelial cells to organize in this direction. Okay? And so that was something that we found to be really exciting. And then furthermore, we can do some additional quantitative analysis to show, okay, well, in fact, you can do certain kinds of image quantifications, including the number of branch points. So this gives you a sense of how many branches each vessel branches off into. And so we can do some additional metrics to compare how vessels form when they're growing on a typical randomly formed scaffold to that of an aligned scaffold. But one of the most important things that we're able to show in this paper is that mainly that the endothelial cells that are formed on these aligned scaffolds are able to better mimic organized patterning of vessels that are better able to mimic, this original picture that I mentioned, where all of the endothelial cells are organized in parallel. So this was just one of the things that we have done in the past to try to understand ways that we can use spatial patterning cues to tell endothelial cells how they should organize so that we can create what we call more biomimetic, or basically engineered tissue that look more like how the native structure should look like. Okay, so I'll skip over some of these images that are not as important. Okay, well that was just an example of how we create microvasculature in our biological scaffold. And so in the remaining time, I want to switch gears to tell you sort of a second side story that's more related to, okay, that's great, but how can we apply this knowledge for something that I mentioned to help save lives, to develop new treatments, to replace to replace organs for donor shortage. Well, even though our lab is a cardiovascular lab, our lab is located at the VA hospital, so we do a lot of work that is focused on the benefit of our veterans. Do we have any veterans here? Okay, great. And so we really care about diseases or injuries that combat military personnel and veterans encounter. And one of them is related to what we call volumetric muscle loss, or VML we call it to be short. But basically, it is characterized by large chunks of muscle that are essentially removed during different types of traumatic injuries, including blast injuries or roadside bombs, even gunshot wounds. All of these types of traumatic injuries can lead to large chunks of muscle getting removed or injured, right? Now, you may say, well, this is all skeletal muscle. What does that have to do with cardiac? Well, skeletal muscle and cardiac muscle have a lot of similarities in terms of their structure and their function. And so a lot of the work that we do that's related to cardiac, we've also applied to skeletal muscle. And so getting back to why we're interested in VML, right, is that most of the current treatments for VML include things like a muscle flap transfer. That means if this part of the muscle is injured, well, you basically extract muscle from another part of your body to place it on the part that's injured. What does that mean? Well, that means the site where you took your healthy muscle, you can have something called donor site morbidity, right? Because you're removing muscle from a healthy region of your body to place it into a region that is not healthy, right? So it's not really the ideal case, right? And this is an area where tissue engineering has promise, because if we can create artificial flaps of muscle, then for these kinds of combat scenarios where we need essentially a replacement of chunks of muscle, we can basically have these engineered muscles as a replacement. And so anyway, so this is sort of what the muscle looks like after it has had large chunks that are removed due to traumatic injuries. And then along the way, we've also incorporated something called rehabilitative regeneration. So not only just implanting an engineered tissue into a region that is diseased or injured, but then using traditional rehabilitative therapies, such as stimulation, physical activity, as a way to see if we can boost the benefit of the engineered tissues that we implant. And this is also a very new area that has just only been recognized in the past few years, the concept of merging traditional rehabilitation to what we call tissue regeneration. And I know that for many of you guys, if you're interested in my slides, I'd be happy to share the slides with you in the end. So, okay, so one of the things that we have applied is we talked about spatial patterning, right? It turns out skeletal muscle, very much spatially patterned tissue. So there consists of basically bundles of these muscle fibers that are interspersed with blood vessels, small vessels called capillaries. And so what we thought is, oh, okay, well, can we create something that looks a little bit like this by creating patterned scaffolds that also have these little bundle-like structures, so that ultimately, when we implant them into mice that have injured muscle and give them rehabilitation, in this case in the form of exercise in a cage view, would essentially the body figure out to say, okay, let's create new muscle, and let's have that new muscle organized based on the spatial patterning cues of the scaffold, so that we can create engineering, so that the body can figure out how to replenish the muscle of its own in an organized fashion. So now in this case, all we're doing is implanting a biological scaffold, no cells, because I'll tell you how it looks like after we implant cells. But in this case, this was just the very first experiment. What if we just gave them a patterned scaffold? Let the body figure out what it needs to do to create oriented muscle. And so we can create, again, oriented scaffolds, similar to what we showed before. And this was our study design. We create this artificial, experimentally induced muscle injury into the tibialis anterior muscle. This is the muscle that's sort of down here in the lower leg, by removing surgically 20% of that muscle. And then we implant into its place one of these scaffolds, either scaffolds that have oriented muscle, or oriented scaffold fibers, or non-oriented, so just a randomly oriented one. And then we took another material that is currently being used in a lot of clinical settings, which are what we call decellularized scaffolds, meaning we take pig intestines, remove all the cells, so it's decellularized, and take that scaffold and say, okay, that's your decellularized scaffold from pigs. Let's use that as a biological scaffold. And then after implanting for 7 days, we then give them the opportunity to have exercise as much as they want in the cage wheel. And then 2 weeks later, day 21, we then try to study how did the muscle regenerate. And the way we look at how the muscle regenerates is that after the study was done in 21 days, we actually removed the entire chunk of muscle, and we look at them in very fine detail by cutting them into 10 micron thick sections. So super thin sections, even thinner than the turkey that you buy at the deli slicer. And then we can do different kinds of stains on them. And one of the stains we did was one called MHC, which stands for myosin heavy chain. And it is a marker that we use to identify newly formed muscle fibers. And surrounding each muscle fiber is always a matrix that's called laminin. So that's basically our way to just delineate the borders of adjacent muscle fibers. And so what was really striking was that in both cases, whether or not on both the aligned scaffolds as well as on the aligned and randomly oriented scaffolds, so that's the blue and green, we were able to get much better or much higher myofiber cross-sectional area. What does that mean? That means the physical cross-sectional area of these myofibers are much bigger, which is a suggestion that the muscle fibers are much more developed and much more mature. But in terms of looking at the animals who got exercise and the ones who didn't get exercise, it didn't seem as if having exercise had any potential benefit in terms of muscle fiber regeneration. But the other element that's really important for muscle regeneration is the formation of new blood vessels. So that's called vascular regeneration. And in this case, it also turned out that both the randomly oriented as well as the aligned scaffolds were much better at promoting vascular regeneration. And then, interestingly, only on the aligned scaffolds, which is the green one, where when we then added exercise on top of that, the vascular regeneration was further augmented from this level to this level. So that gives a suggestion of, oh, okay, it seems that the addition of exercise has some benefit on improving blood vessel formation. What about nerve formation? Well, we looked at the nerves using a marker that's called alpha-1 protoxin, which labels what we call the neuromuscular junctions. And the more of these junctions means that there's more innervation. And because there aren't that many of these junctions, we looked in a periphery of 500 microns radially from the site of the scaffold implant, as well as 1,000 microns radially from the site of the implant. And it turns out that on our aligned scaffold with exercise, which is what the black bars are, that group had actually the most number of neuromuscular junctions, suggesting that these aligned scaffolds were able to improve the innervation of the tissue. Okay, so that was the initial study, no cells, just purely looking at spatially patterned scaffolds. What do they do? In the remaining few minutes, I'll just give you a few punchlines of some of the things that we learned when we then put in cells. So what I mean by that is we figured, okay, scaffold alone is probably not going to be good enough. Maybe we have to actually create engineered tissue, which means having the actual muscle fibers as well as the blood vessels. And so we seeded these scaffolds with both myofibers, we call myotubes, as well as endothelial cells, and then did the implantation. And this gives you a sense of how they look before we implant. So the myofibers, before the myotubes, are stained by myosin heavy chain, shown by green. And then the endothelial cells are shown in a reddish color called C31. But the thing of interest is that on the aligned scaffold, those myotubes do develop along the direction of the scaffold fibers. And so they really can sense the spatial patterning cues of the scaffold. And this is just a movie, you can't really see it too well with this light, I guess, but what I was trying to show you is that when we stimulate them electrically, engineered tissue contracts synchronously. So that's something that's kind of neat about muscle. You can stimulate them and then they will contract. Okay, so then we did the actual experiment and then implanting the various groups, so just no cells, or you have both randomly or aligned scaffold with just the myotubes, or in co-culture with the endothelial cells that form the blood vessel. And I'll skip to maybe some of the things that I think are most interesting, which is that we then looked at, okay, did we get improved muscle regeneration now, right? And so as I mentioned, we were able to modify the cells with sort of GFP from the jellyfish, right? And so in this particular study, we labeled the myotubes, the muscle cells, with GFP. So all the GFP positive ones, the green ones, were all derived from the cells that we transplanted. So that's how we're able to see that, oh, those are the ones that we transplanted, the ones that are green, right? And so we're able to find the number of these green positive muscle fibers and show that, hey, when our scaffolds were aligned and seeded with both the myotubes as well as endothelial cells, so this is what we call engineered endothelialized muscle, the number of newly formed muscle fibers was actually the highest here. And importantly, I'll just skip some of the highlights, is that, okay, what about the vasculature? Now what's neat is that these are all tissue sections. These are biological tissues. And what we can see is that in the animals that we treated with the aligned scaffolds, we were able to find that the vessels that were formed in the region were much more aligned, organized, compared to the ones that were on the randomly oriented scaffolds. So this gives you a sense of how it looks. This is sort of a 3D reconstruction. So these are the transplanted myofibers, so they're expressing green because they have that GFP protein in them. And then these are the blood vessels that are labeled in red. And isn't that kind of cool to show that our implants are able to organize the formation of muscle structure such that we have a sort of organized pattern structure that is typical of native muscle. And so just to summarize some of the tidbits that I mentioned today, I wanted to summarize by saying that we can use spatial patterning as a way to guide the formation of vasculature. And hopefully, the idea is that we can use spatial patterning to create new engineered tissues, including new blood vessels, as well as new skeletal muscle, as well as new cardiac muscle. So this is a little bit of some of the research that I wanted to talk about. In the interest of time, I know that some of you guys will need to leave for various reasons, and that's totally fine if you need to leave. I was also asked to give some words of advice. And so I wanted to just leave this talk with just 3 pieces of advice. So what's the first piece of advice? I would say is trust that college is a transformational process. What does that mean? Many of you guys are freshmen, right? And you think like, okay, everyone told me college is going to be great. I'm going to go to college. I'm going to pop out of college. I'm going to be a different person. I'm going to be marketable. I'm going to be someone that companies want to hire. And many of you guys are seniors. You're like, oh, I've been here for the past few years already. Did I really change? Did I not? Right? You may be questioning that. I remember back when I was a college student, I thought, oh, okay, I spent all my time just trying to work hard in classes, and suddenly I'm like, okay, so am I a different person now? What makes me now, as a senior in college, different from back when I was a senior in high school? Am I really that much better suited to be hired by companies? Well, my response is I trust that college is a transformational process. It's not necessarily about, okay, how much you've learned just from the classes you've taken alone. A lot of things have changed. You've matured, the way you think, your knowledge about the world, your worldview has all changed. And in the course of the different classes, beyond the actual contents of the work that you've learned a lot of life skills, like how to adapt to stress, how to deal with deadlines, how to deal with failures, how to negotiate, how to work in teams. And those are all important life skills that you need to be successful at the job that you do in the future. And even though it's not strictly based on the content of the class that you take, but this is one of the things that are a byproduct, a good byproduct, of going through the college process. The second or the third advice that I would give is develop your passion. Now, what does that mean? Many of you guys have maybe heard of the phrase that says, follow your passion, right? And do what your passion tells you, right? Now, it's really recently been appreciated that passions are dynamic. Some people may not have a passion right now, and that's fine, right? And maybe you do have a passion right now, but your passion 5 years from now maybe is different from what it is currently. So your passions will change, and maybe your passions will grow, and your passions will shrink, that's fine. But generally, your passions usually lie in the intersection of what you like, what you love, the things that you're really good at, their skill sets, and the things that you find to be important or values. And usually the middle is sort of an area where at least you begin to start looking for your passions. And if you don't have if you can't identify your passion right now, that's okay. It's going to evolve, right? And sometimes you may depend on focusing on the things on the outside, focusing on the things that you like to do, the things that you're good at, the things that you find important. And eventually, through these various life experiences, you'll fall into that sweet spot of where the things you find passionate will become obvious to you. And so I think that developing your passion is more important than specifically your major is. Right? I think that many of you guys, when choosing your major, may have had a block to say, oh no, if I choose the wrong major, my life is doomed. And let me tell you that your life is not doomed even if you choose the wrong major, right? Because as I mentioned, the college process is not just about the major that you are in, but it's about all those other life skills that you learn through the college process that is the same no matter what kind of major you select. And finally, the last advice that I'll give is to seek out mentors, right? Back when I was a high school student and a college student, I really relied on mentors who basically had my best interests, and mentors who I was able to ask questions, mentors who were able to guide me in the right direction, to really work on developing the passions that I was having at that time. And so I would say seek out mentors. Mentors are important. Mentors could be anyone. They could be people who are your teachers or professors. They could be people in the neighborhood, in your community that you look up to. They could be family, friends. And what's most important about the mentors is that you want it to happen organically, right? You don't just walk up to someone and say, hey, can you be my mentor? And someone goes, okay, and then poof, I'm done. It's not, if you don't already have existing members that you see like day in, day out, that kind of thing, one of the things I may ask you is to think about who you find admirable in your life, right? Who do you find that are people that you admire, that you want to be, right? And figure out, okay, what is special? What qualities does that person have that make them admirable, and what kind of qualities you would like to emulate, right? And from there, you know, if it's not someone that you typically see on a daily basis, like let's say one of your teachers, you can informally ask them for like an informal meeting over coffee or something, right? Where you want to have questions lined up that you want to get advice on, right? But importantly, these mentorships come out very organically, right? So I would say, seek out mentors, and you can have multiple mentors. Maybe some mentors are good for career advice, whereas other mentors will be good about grades, and others will be good about family and personal issues, right? You can have multiple mentors for different kinds of things. But the idea is that these mentors will hopefully guide you in the right path for your future success. And so with that, I'd like to just thank my lab members, and you can always follow me on my Twitter handle and our lab website. And I'd be happy to take your questions. Thank you. Yeah, so I didn't have the time to go into a lot of detail about it. But we're using microgravity, as a way to study muscle boosting movements, such as sarcopenia, muscle atrophy. So that's one of the applications that we're using microgravity.
Just want people to
Right, so it's not related to that, it's actually availability. If we had an infinite number of like donor kidneys, donor hearts, donor everything, then maybe we wouldn't need a tissue. But the fact is that we don't.
Is there any knowledge on that?
Right, so the idea ultimately is that we would create engineered tissue derived from ourselves so that we would not have rejection. So what I mean by that is, I talk about these inducitory stem cells, these iPSCs. Those are stem cells that you generate from yourself. So you can take some blood, you can take your skin cell, and convert that into your own iPSCs. And then turn them into whatever cell types you want. So the idea is that then you basically have this bank of your own cells that will not reject you. Right? So you use that to create your own personalized engineered tissue that you can kind of adapt to yourself without rejection.
So stem cells have to come from a particular patient to regenerate their
Exactly. And so we envision is a day when, like, in the same way where, you typically, let's say when you're born, you do all these like APGAR tests and this and that test. Then the standard routine thing would go, you have to extract some blood from the baby and then basically create iPSCs. Everyone would have an iPSC bank somewhere, so that whenever it's okay, we need it for for disease, or we need it for tissue, then those cells will just be used. That's the idea.
Thank you.
Are we there yet? No. I guess.
So what have because in the transfer last year, you have been an engineer, have we been successful with any patients so far?
So most of the work are still in what we call clinical stages, so we're just like small animals as well as larger animals like fish or sheep. There are a number of hurdles that we have to encounter, so as brought up about tissue rejection, right, which is that if there's not cells from the same person, they will potentially be rejected unless you give some type of immune suppressant. And sometimes it's tricky because we're testing them out in all these animals, right? But these are human cells, right? We're trying to create human tissue, right? Not pig tissue or mouse tissue, right? So we put human tissue in these animal models, right? The only way that will work is if they are compromised, right? And so that's one of the limitations of our studies, that we can put them into animals, but they all have to be immune compromised, right? So we've actually never done a study where we put actually human cells, a person's own human cells, in that configuration. One of the caveats that I mentioned with these iPSCs is that although they're very powerful, they do have some different kinds of hurdles through the FDA approval process. So in the US at least, there are no therapies right now that involve iPSCs that can advance to clinical trials. Now currently, in Japan and other parts of Europe, they have initiated clinical trials to determine if iPSC-based cell therapy is safe or not. And so I would say that Japan is leading the way right now. At least in the US, due to these regulatory hurdles, we don't have that, basically.
So like on the Cell Molecular PRP, like that's like a very basic form of that, right? So, that's not, like, it doesn't produce like enough stem cells in order to like, to go forward with any
Right. So I believe the PRP, which is sort of plasma, right?
Right.
So those are not considered, those are biological, but they're not cell-based. So that is basically a protein-ish. So PRP will not have the same kind of rejection issues that you would if we had actual cells that were infected. So I think that's why cell therapy is a lot trickier to do compared to like plasma.
And also, so there's a lot of rules as well if people, right, some of them are very afraid that when it's especially when it comes to stem cells or whatever you're doing.
Right. Exactly. So the thing that the government used to be very afraid of, so imagine like George W. Bush, in that era of his presidency, he made a very critical decision regarding embryonic stem cells because of the fact that embryonic stem cells are derived from embryo and they have a lot of ethical issues. The nice thing about iPSCs is that they're derived from any kind of random cell in your body, so they don't have any kind of ethical issues, so that's the reason why iPSCs are favored over embryonic stem cell research. But the issue is that because they're so flexible, these pluripotent stem cells are so flexible, while we can tell them, okay, turn into this kind of cell, right? The main concern that the FDA has is what if you implant them into the body, and then suddenly the body says, oh, turn into some other kind of cell, right? So there's that uncertainty of what happens when you implant the cells in the body. Will the body tell them to do something unless they can coax them to turn into cancer, for example, right? And so that uncertainty has been one of the limiting factors for why the FDA is still waiting for more data to really confirm the safety of these clinical cells, which means doing primate studies. But in the end, all of those studies are not the real thing, right? Because even in the primates, they usually will get immune suppression, which means, okay, well, if they're immune suppressed, they're not acting normally like a human body would, right? And so other countries, including Japan and other parts of Europe, have been a little bit more progressive in that. To say, like, well, as long as it doesn't cause cancer, let's try it, right? So there's enough safety data from other countries that suggest that these iPSCs and cells derived from iPSCs are safe enough for at least a small yes.
So during research project, how do we make sure human body will not reject engineered tissue?
Yes. So it's getting back to that question about the rejection. So the theory, the goal is that ultimately we would create engineered tissues that are not rejected because they come from your own cells. And that's what these iPSCs are. So they're cells that you can basically take a skin cell and basically genetically turn them into what we call iPSCs. And once they're in this iPSC state, they can turn into whatever cell type you want them to be, including heart cells, right? So if you want to put in your heart tissue, you would take your own iPSCs and make them to whatever engineered tissue you want and put them back inside you. But in order for this whole thing to work, there's really two possibilities. Either we evolve to the point where everybody has their own iPSCs bank somewhere, it's like, oh, it's my back pocket, here it is, right? Or the case, and biologists, computational biologists have kind of calculated the numbers, which is that, as you know, it applies the same way to organ transplants, right? The only reason why organ transplants can work is that there are some kinds of organs that have certain kinds of signatures that you are compatible enough with, so that if you took that person's organ and put them inside you, you would not have a major reaction. You have a minor reaction, but not a major reaction. And the same idea goes with iPSC, that maybe we don't need everyone having their own iPSC line frozen somewhere, but that if you generate enough diversity of lines, then among those number of lines, generally speaking, most people would be able to use at least one of these lines for them. And so, to give an example, I think one group had estimated that for countries that are very homogeneous, like as in like genetically homogeneous, like mostly from the same, race, for example, it's estimated that I think you need something like 300 or 400 lines, and those 300 or 400 lines would be compatible to match up to 80% of the population. So meaning those 300 lines could be used on the majority of this 80% of the population without rejection. But now for countries that are very diverse, right, then you would need many more lines than that, right? And so that's been another, argument basically. So either everyone gets their own, or for countries that are very much more racially homogeneous, you would only need a certain number of lines that could be tolerated by the majority of the population. And then getting back to your question about bone marrow stem cells, right? I haven't talked anything at all about stem cells that are found in the adult body, so what are called the adult stem cells, right? And there are a lot of stem cell trials in the clinics right now. We're using adult stem cells, meaning those from bone marrow, those from fat, for example. And for the most part, those results have been mixed, in part because of the fact that it's pretty well appreciated at this point, because we've done these kinds of adult stem cell trials for at least 10, 15 years now, is that those cells, one, don't really stick around, two, they're not very flexible. So if you told, bone marrow stem cells turn into a heart tissue, not going to happen. But if you tell the bone marrow turn into a blood cell, like, oh yeah, I get that, right? So meaning that bone marrow stem cells can turn into certain kinds of cells that seem to make sense, like because bone marrow naturally makes, blood, for example. Like, oh yeah, blood cells, yeah, they can. Those bone marrow derived cells, yeah, they can turn into blood cells easily, right? So, but if you tell them, oh, turn into a heart tissue, not going to happen, right? And so, because of that, that's one of the limitations with adult stem cells. The second is that they don't survive very well. So in most cases, when they've injected adult stem cells into the body, for the most part, they only survive for a short period of time, which means that they have a small window of opportunity, and because they're not turning into any cardiovascular lineages, what are they doing, right? And the idea is that they are producing chemical factors that may still be beneficial to the tissue, even though they're not turning into the cell type that the tissue needs, but maybe they're at least producing chemicals that are good for that tissue. So that's really the idea of what adult stem cells are. That's the general consensus of what adult stem cells do. Any additional questions? Yes.
Yes, Doctor, I saw something on 60 Minutes, and it was rather fascinating. I don't know if this is in your area of expertise, but NIH did a very experimental trial using genetics, DNA, for sickle cell anemia. And basically they came up with a way of changing by feeding an egg. And this person had sickle cell anemia, and they basically had to treat them with radiation, and then they injected them, I think, with new stem cell DNA. That person now has no sickle cell at all. Are you aware of that?
Yeah, so this is...
I mean, it was fascinating.
Right, so this is all going on in parallel to the work that we do as tissue engineers, right? Because ultimately, that's the question that we have is, we're trying to create engineered tissues for a patient. Then there are patients who have certain type of genetic abnormalities, for example, right? And when we engineer this tissue for the patient, do we correct the genetic mutation, for example? So that's sort of a question that we have to think about. But getting on to your question about sickle cell anemia is, this kind of genetic modification is really limited to diseases that are characterized by very specific genetic mutations, right? And that is fine. So I think that is a neat way, basically, to do what we call gene editing as a way to treat diseases that are known to have very specific mutations, right? At least in the cardiovascular research world, right? Most of the diseases that we deal with, like heart failure, which are driving heart attacks, atherosclerosis, strokes, those kinds of diseases are not purely genetic. In fact, there's no specific genetic abnormality that one can even identify. And so because of that, we couldn't do gene editing because there are no one genes, right? So the idea is that there are probably thousands of possible gene modifications that could contribute to the disease, right? And then secondly, then there's the lifestyle factor, right? So genetics are one component, but for most cardiovascular diseases, it's also lifestyles. So both of them come hand in hand. And so I think the bottom line is that gene editing doesn't work for what we call complex genetic diseases. So we think that most cardiovascular diseases have a genetic component, but they're not a simple, switch an A to a T and boom, we're done. It's not as simple as that, unfortunately. But there are diseases, including that of like sickle cell anemia and other kinds of diseases that are really one specific mutation. So if you can gene edit it, the disease is cured. And so I think that has its place in certain kinds of diseases with those really simple mutations. And then for most diseases which do not have a simple mutation, we're still left with the same issue that gene editing will not be applicable. Great, well thanks for coming to my talk. You can also follow us on Twitter.